going to be really interesting to hear the latest on quagga mussel research and potential for control. Dr. Ashley Elgin will give their talk, and she's from the Lake Michigan Biostation on Muskegon. You want to talk a little bit about your background?
Yeah, sure. And I know, don't hesitate to get up and get some food because I'm just kind of starting out with some general background. So feel free to walk around while I'm doing that, just intro into my talk, just in the interest of time and get us going. So yeah, I'm a research ecologist with the NOAA Great Lakes Environmental Research Laboratory. I'm based just down the coast at our field station, our beautiful field station in Muskegon. So I get a firsthand, I can look out my office window and see how many people are fishing that day. I can see where people are pulling up a lot of drum on the pier. So I have a really good seat to watch all the fishing activity. And then, but what I think about a lot, I've studied these for over a decade. I've studied invasive species for more than 20 years, is the quagga and the zebra mussels. So that's what I'll be talking about today. Last time I presented for the Ludington workshop, it was late January of 2020. So things for humans changed a lot, starting right out since that last time I presented at this workshop. If you're a mussel, things haven't changed so much in that time. But from a scientist perspective, we've learned some new things about mussels, and we're putting more efforts towards learning how to control them. So in this presentation, I'm going to be focusing on those aspects. First I want to, these are my co-authors here. I work closely with them on what I'm presenting here and many of my current projects, but I also have many collaborators across multiple state, federal agencies, academic partners. So I just want to show this work's only possible due to all these partnerships. So in the overview, I'm going to kind of ease in with background about dry-seeded mussels, talk about their population trends, and spend some more time at the end on control options and where we're at with that. Some common themes I want to help you know over the course of this talk is that depth really matters for mussel biology. There are aspects of mussel biology that make them really, really difficult to control. So I'm going to be working that in throughout just to help you know more about mussels and why they cause so many problems. So I'm talking here about two species. I'm sure you're all familiar with both of these species. You have quagga mussel on the top and zebra mussel on the bottom. Zebra mussels came in first. They made a big splash. They made a big impression on people. People were seeing them attached to everything. But then quaggas came in after, kind of quietly, but they are much more abundant. And as a result, they have much higher impacts in the Great Lakes. Their general impacts are that they reduce the spring diatom bloom. So that top mussel, it has that big siphon extended out. And sometimes I have pictures of siphons that'll be almost the full length of the body. They put the siphon out and they will relentlessly filter the water. And as a result, all the water is passing through a mussel. So in the springtime, when the water is fully mixed, all of the water in that water column has an opportunity to pass through a mussel. It filters it out. It takes the beneficial spring diatom bloom, it knocks that down. NOAA research has shown very clearly how much they knock down the spring diatom bloom. In the process of removing all the phytoplankton from the water, they're increasing water clarity. And think about how that changes the relationship between prey and any visual predators, because now that predators can see further. So it affects that predator-prey relationship. Ultimately, you're having energy shift from the water column and being entrained down in the benthic zone on the bottom of the lake. And so, Ben had touched on this with what's happening with alewife. Well, they have less to eat. They're now it's like growing in a sand patch versus a good loam. So they just take all this energy and they trap it down in mussels on the bottom. And I have some great pictures to show you what that looks like. What's the reality on the bottom of the lake bed? What does it look like? Other impacts that they have that you may be familiar with is since they attach to things, they can displace native mussels. And that's shown here. And then they also can foul infrastructure, boat props, docks, anything that's a hard surface, they will cover it if they're present. But I think I thought was what important to this group in particular is talking what's happened to the benthic, the bottom dwelling community over time and the other organisms. So I've represented this figure in two different ways. What the benthic community looked like by the major taxonomic groups starting in the reference here is 1980, and then what it looks like now. And so this line graph shows over time, color coded, where diparia are in green. I'll highlight them, very beneficial native amphipod, great food source. I've called them the Snickers of the deep because they're high in calories, high in fat, and good quality. If a lake whitefish is lucky, its guts are full of white diparia. So in 1980, you can see at the beginning of the graph, and also this first pie chart in the upper right, that they were the dominant benthic organism in 1980. I'm just going to fast forward now to more present times in 2015, and it's that lower pie chart. And now the dominant benthic organism is the quagga mussels. And the number 2 in both cases is the blue, and those are oligochaetes. So those are another common fish food, and oligochaetes are always second place. They've been consistent over time, but what we've seen is a shift from diparia dominance down to mussel dominance. And I put some, if you're interested in the details on here, the first star shows that zebras arrived first, and then quaggas arrived. Now diparia declines, they became coincident with pretty much when zebras arrived, and it just continued after quaggas arrived. So I have some graphs to show you what that looks like in a different way, because I know that this graph, okay, you get things went up and down, but I like to see things spatially. So we produce what we affectionately call our blue density graphs, but we all know what that means. And what you're getting is you get a darker blue color that is higher density. And then you get to see the patterns of spread. And what I like here too, you get the play between zebra mussels coming in and then quagga mussels. So zebra mussels came into the lake, dotting around the perimeter of the lake, with a focus kind of central and gain up to the north, between 1995 and 2000. And then in 2000, other thing that is notable that's happening, if you look in the north, that's the first time that quaggas were arriving in the lake. They started in the north, and the difference between 2000 and 2005 is striking to me because they managed to get just tickling up in the north to the entire perimeter of the lake. So here's the first key point about mussel biology is they spread by a planktonic veliger stage. And so it's external reproduction and fertilization. It creates this veliger, and that veliger can be in the water column for quite a while. And during that time, it will circulate, it'll distribute around the lake. And that's how 5 years makes that much of a difference in the quagga mussel population. And zebra mussels do the same. But another thing I want to point out that by the time quaggas in 2000 or 2005 are quickly colonizing the lake, zebra mussels are already waning, and they are going down in numbers. And by 2010, 5 years later, we stopped getting zebra mussels in our survey. So quagga mussels completely displace them. So then the bad joke is, how do you get rid of zebra mussels? You introduce quagga mussels. We were successful at that, and now that's essentially all we have. And these are in the most shallow sites here, about 15 meters. And so really, we don't find zebra mussels except for 1 or 2 a year at a few sites. So it's a quagga story, and a lot of these impacts on the whole lake scale are because of quaggas. To keep on this theme, I also want to share, this is what's happening with diparia at this time. So 1995, these are the densities. I don't have a figure for before this. Their densities were already declining by 1995. But then they were just rapidly, and they continued to decline, and that there are a few holdout populations in the central and the north part, but diparia are way down. And that just has major implications for the quality of diets when you replace diparia, Snickers bars with quagga mussels. And I don't know what's a good, I don't know, I don't know, potato chip, I think I've heard that. Hair,
The
yeah,
nutritional equivalent of hair.
hair, because it's so much of the shell, you have very little, it's, 10-to-1 shell to tissue weight when you dry them out. So another way, yet one more way to look at this at biomass is it's gonna go back to a line graph, but it's gonna spread it out by depth, because that's another theme here, is that depth really matters for mussel biology. It explains their population trends, and I think it's important for when you think about controlling them. So in this graph, I'm going to be putting out one line at a time, going from shallow to deep, and you can see what's happened over time. So this is this is just looking at that. This is quagga mussels only, just focusing on quagga mussels, and now I'm reporting on biomass. So in the shallow zone, it's a high energy zone, shifting sands, lots of storms, and it bounces up and down year to year. There's no boom or bust here. It just bounces around, highly variable. And we see that here. But then compare that to the next depth zone, 31 to 50 meters, and rocket it up. This is just, talk about a major boom in population. And it came down, not totally a bust though. It came down to be about half of its peak, and it's bounced around ever since. And these data go up to 2024 is the most recent data we have analyzed. People say, oh, that's great, they're cut in half. Well, at that level, this lower level, they still have extreme impacts. And just anyone out there think about the impacts we've seen on the food web, even over the last 15 years, they haven't reduced because that level is enough to cause impacts. The next line here, going a step deeper, is similar pattern. It took longer for the population to develop and hit their peak, and then they came down, but it wasn't as much of a crash. And now they're bing bong, bong, just going along. So then last, we'll focus on the greatest depth zones, greater than 90 meters. Now this is an important depth zone because most of the lake, this is greater than 90 meters for Lake Michigan. It's the highest proportion of the bottom. And it took them a long time, and they eased up to a steady level. They didn't overshoot it. They just gently got up to that level, and now they've been going back and forth. So it's just an example of it's like at these three depths, the mussel populations are traveling at different speeds. And then that determined how much they overshot what the lake could hold. So now we're at a point where at all depths, you see actually, they're not too far apart from what the biomass is at each depth zone. And when we put this together in this thick back line or a weighted mean, it's been quite steady since 2010. So I told you and now I'm going to show you a fourth way what this looks like. This is what it looks like at about 50 meters. This is what a carpet of mussels looks like. So you have some open areas. That's just solid mussels. And that would, if you were to look at this, and in one of this is our Ponar grab, our steel claw that we send down to get bottom samples. You'll have thousands of mussels in one single grab. It's going to be 15 to 20,000 mussels per square meter is what it comes out to. And then probably it's even a better view here, just to get a sense though of this is how dense they are at this depth. When you go further offshore, which is even harder to get pictures of, we have these great images of this is what they look like. The mussels are pale, they're very slender. You can see that their siphons are way extended, and they are able to be in soft sediment, which is unique for quagga mussels. But they have these long siphons so they can outcompete their neighbors and get that food before their neighbors do. But this is it's like a regular distribution, even though it's not as dense, but they're clearly successful and they are there. And you go to the deepest areas of the lake, you think they're not there yet. They're there. They're just a little more sparse. So keeping on this theme of depth matters, I'm just going to express some depth differences in different ways. So the first one, we saw that was a 35-inch Chinook is a certain weight, and that's one way to look at a standard weight. Well, here we have a 15 millimeter mussel, so it's about a 9/16 inch mussel, and this is standard weight of it. And the main takeaway here, I have several years of data. but I just want to focus when each of these clusters of bars is a depth. And at the deepest depth zone, the mussels are relatively heavier than all of the other depth zones. And then the other, another way to, so that's what's, that they're just heavier in the nearshore zone. And that's because they're in warmer temperatures, they're in more productive waters, and that supports more growth. So based on a study I did in Lake Ontario, we see very clearly that, and here again, it's that light blue is shallow and darker blue is getting deeper. Shallow mussels grow a lot more than mid-depth mussels, and they grow a lot more in a given year than, or in a year's time, than a shallow or the deepest mussel at 90 meters. There's actually they grew 10 times as much at 15 meters than they did at 90 meters. So one common thing I get, someone will hand me a mussel, how old is this mussel? They're like, well, where did this mussel come from? Because you could have a 25 millimeter, that's an inch long mussel. It could come from the shallows and it could be 2 years old. Or you could get 25 millimeter mussel and it comes from 90 meters and it could be 2 decades old. So it just shows how long lived these mussels are, and probably more so than you think. And it shows that they would have slower population turnover depending on the depth. And then another very important thing, if you want to think about what mussels would you target, who's contributing to all this new growth and recruitment, is you want to look at their reproductive activity. And this is some very preliminary stuff that I have, and the weather's bad tomorrow. If the weather's good on Wednesday, we're going to collect mussels and add another cluster of bars onto this graph. It's going again, light blue is shallow and darker blue is deep. A higher bar here shows a greater percentage of their body weight that is in reproductive material. And in different seasons, this is July, September, and February. July being a peak reproductive time, you have twice as much proportion of their body is in reproductive material for a shallow mussel than a deep mussel. And it fits into the story of shallow areas, more energy is there for them. The mussels are heavier, they grow faster, they have more energy to put towards reproduction. So just a very important difference of where the mussels are and how they're behaving, and what you can expect from them, that population maybe 10 years from now. It very much depends on depth. Okay, then from reproduction, we're just going through the full life cycle. That brings us back to veligers. I mentioned they have this planktonic veliger stage, and that's how they can travel around and colonize a lake so quickly. So my colleague, Steve Pothoven at NOAA, has been doing a lot of work on veligers for many years. And he's found that in general, we see synchronized peaks at our 3 study depths off the coast of Muskegon, and that these peaks in veligers show that there are 2 spawning events that occur in Lake Michigan every year. And it kind of flexes around, and some years one of the events might be somewhat muted, but we see this general trend of a somewhat coordinated spawning event at all 3 depths. And one reason it's coordinated, we think, is that the mussel, the veligers could be produced maybe more in the nearshore where there's more activity, but then it distributes with the currents, and that's one way that you're getting veligers further out offshore, but they may be coming from nearshore. And we're continuing to do work to really try to pin that down and demonstrate that. Another thing that's important to know about veligers is just the main signal I want you to see in these bar graphs is that high years and low years, kind of cyclical, high and low years for veligers. When veliger is in a high production year, as you can see that I highlighted with red there, is in 2023 in the nearshore, they can be a very major component during times of year of the plankton biomass. So if you're a larval fish at that time of year, and larval alewife are going to be very present when veligers can be very common in the water column, and that could be interfering with the food you want to eat. Because an important that third point is veligers have a low energy density. It's going back to the whole, I guess, Snickers versus a ball of hair analogy. you could compare that too for zooplankton compared to veligers. Veligers have much lower energy density. Steve Pavlovan and I have seen this. We put it, well, last summer I collected a pure enough sample of veligers to put it in a bomb calorimeter and get the calories of a cluster of veligers. And it was about 1/3, 1/4 of what you get in that same weight of zooplankton. So it just shows that in the same way that settled mussels are a worse food source, veligers are a worse food source for larva fish and other that could be feeding on them at that time. So to round out here to talk about control options, what can be done? I get that in every talk. What can be done with these relentless, constantly filter feeding, easy spreading species? And to address this, the Invasive Mussel Collaborative was formed over 10 years ago in 2014 to look for an advanced scientifically sound technology for control. And since then, many toxicants and physical treatments have been developed and they're actively being tested. So I'll do just a quick rundown to show you there's a wide variety of approaches. There are toxicants such as potash, niclosamide. So instead of using it for lampreys, let's use it as a molluscicide, and it's approved for that. Copper compounds are very lethal to mussels, but all of these toxicants, these first three that I mentioned, they're lethal to other organisms as well and other inverts. So then another product was developed, and it's Zequinox. It's from a heat-killed bacteria. That's very species specific, that it'll be lethal to quagga and zebra mussels, but it won't be lethal to native mussels. It just has a different mechanism, and it's lethal specifically to the quagga and zebra mussels. So that shows just a breakthrough in finding a toxicant that's very species specific and that's difficult to develop. There are physical and other treatments I'll just go through quickly. Benthic mats are used for smothering mussels, and they can be used. This picture is from a boat launch in Texas, but just to show that you can smother the mussels if you find them quick and you can get on top of an infestation. You can use carbon dioxide. I have a colleague at USGS who's incorporating putting carbon dioxide under mats as a way to cause quick mortality. Some areas can do water drawdowns, but with a lot of these, that's difficult to implement in the Great Lakes because the mussels are found so deep and there's so much area to cover. So you can just see that some of these tools would be easier or more difficult to scale up than others. And then I'll spend now a little more time on physical removal and destruction, because this is an area where NOAA has been doing research over the last few years. We developed this robot that is, it's using an underwater crawler. It's a tracked remote operated vehicle. And this year we're going to be testing it out more in an autonomous mode. And then it has a custom designed implement on the front of it that brings the mussels up on a conveyor belt, come over by an auger, and they go through a crushing mechanism and then get left behind. So it's more of a proof of concept study, just to say, could we get an underwater vehicle that is down there doing the work? Because if you could get power to it, and if you could, the big challenge here is it leaves behind a path of destruction, but you want it to be most lethal to mussels and not lethal to other invertebrates. And those are the aspects that we're working on tuning in this pilot study. But here's another view of what it looked like in the path behind where we operated this ROV. You could see the cleared path through that carpet of mussels. Another aspect, anytime you touch the bottom, you create a sediment plume. And so another thing we're working hard to try to limit is the amount of sediment disturbance, which is hard. It's hard to avoid that anytime you're working on the bottom when you have fine sediments, as you know. I'll also, as I conclude here, just highlight a project, the Great Lakes Swim Project, which stands for the Spawning Whitefish Invasive Mussels Project. They're assessing if you can do localized experimental removals of mussels on a high quality habitat, which first thing that comes to mind for many people is a fish spawning reef. And so if you do that, remove mussels from a fish spawning reef, can you actually improve lake whitefish production in a way of improving embryo survival? So Ben is very involved in this, and if you have questions on the whitefish aspect, he'd be able to answer that. And it's a collaborative effort of many agency, tribal, academic partners. It's a multi-year project, and I feel like we're just getting going on it. We've been planning for years, but we've been doing some pre-monitoring, control tests, and now we're getting ready to ramp up more in the next years. So to wrap up here, I'll say that Lake Michigan quagga mussel populations have been relatively stable. Depth plays a major role in, what their population trajectories have been and where they could go. The mussel biology, though, has challenges for control, largely because mussels have not, we haven't found the Achilles heel for mussels like we did for lamprey. Because mussels don't retreat back into tributaries as part of their life cycle. Their life cycle is their advantage because that allows them to you clear them out, and it could be within a year, you have a whole new stock of mussels coming from an untreated area to repopulate where you just treated. So that is one of the major challenges for mussels. The control tools that are under development, they just require extensive work and continuing innovation. And then the control efforts that we're putting a lot of effort and funding from Great Lakes Restoration Initiative is going towards are looking at this localized high value habitats. And we are a ways away from having something that you could apply lake-wide or even on a regional scale. So with that, I'll say thank you very much. My email's here if you just want to follow up later, if you think of a question later. And I'm happy to take any questions, and thank you for staying late.